Welcome. I'm Leslie Cooper, and for the last 20 years, myocarditis has been the focus of my clinical interest and research. The reason I study myocarditis is because a young woman who I cared for when I was a medical resident died of giant cell myocarditis in the coronary care unit, and I'd been with her during the days before she died, and there was nothing we could do. And in those days, there was no treatment. It was a 100% fatal disease. And that bothered me a lot, and so that led me on this path. And here we are 20 years later, and with many good friends. I share in your tragedy. Everyone here has lost someone. And so this group, we're here to support each other. We're here to make advances, to educate the public and physicians, and to ultimately cure myocarditis. Now that's a long-term goal. What we're doing in the interim is trying to make it better for everyone who has to be impacted by this disease. What I want to do in the next 20 minutes is give a short overview of the global burden of the disease. There isn't one answer to what is myocarditis, the topic of this talk. You all know well what it is. You've lived it. You've experienced it from your own experience. One of the problems that we have studying the disease is that it was named by a pathologist 150 years ago as a finding under a microscope. But you all know it's not a finding under a microscope. It's really life and death and heart failure in the real world. And if you're an imager today, it looks like maybe a white spot on a cardiac MRI. If you're in the laboratory, it could be a blood test or a pattern of blood tests. If you're a pathologist, it still looks much like it did 150 years ago. It depends on what perspective you're coming at on how you define this disease. And that's one of the great challenges. But to get a handle on it, up until the last couple of years, no one had ever tried to estimate the global burden of myocarditis. How many people on Earth die of myocarditis? How many people have heart failure as a result of myocarditis? One of the great challenges when I took on the global burden of disease study for myocarditis, this is the major paper, there's a whole issue of The Lancet that was published last year, looking at all diseases, not just myocarditis. But what we learned, the team that I led in this process of reviewing all of the available information in the world, was that because the vast majority of the world, Africa, Asia, South America, have no access to heart biopsies and very little access to cardiac MRI, the diagnosis is very rarely made there. Even in North America and in Europe, where we have access reasonably easily to these technologies to make the diagnosis, many people are diagnosed at the time of autopsy or heart transplant. The diagnosis is not frequently made during life. And so we realized there's only that we had to combine myocarditis with what it looks like usually on an echocardiogram, dilated cardiomyopathy. And that's one of the more common presentations with heart failure of myocarditis, but it's not the only one, obviously. This doesn't include sudden death. And when we combined those two, we were able to come up with the estimate of about 400,000 deaths worldwide for myocarditis and dilated cardiomyopathy. If you think that a reasonable estimate is that 20% of the dilated cardiomyopathies were directly related to myocarditis, it could be as high as 50%. Some of them were familial or genetic. And so somewhere around 80 to 100,000 deaths worldwide occur from myocarditis. Now, 60% are in men, 240,000 roughly, and about 40% are in women. And the reason that myocarditis and dilated cardiomyopathy are more severe in men and cause more sudden death is the subject of research that Katelyn's going to talk about. We're actively working on that to figure out what the triggers are for arrhythmias. I'll talk about that in a little bit. But this is the picture. This does not include sudden death. It took 6 years to gather these data just on heart failure, and we have yet to incorporate sudden death into that model. If you look at it from a different perspective, which is how much of the universe of heart failure is related to myocarditis. And you go, and we looked at about 6 million hospital dismissal summaries, and you can see that if you just look at what the doctors said was the diagnosis, about 3% of infants and about 1.5% of young children, almost 4% of young adults, and all the way out to age 80, as far as we went, there's still about 0.5% of heart failure that's related to myocarditis. The vast majority of the hospitals that reported these data have no access to MRIs or heart biopsy. And this is undoubtedly an enormous underestimate of the true burden of disease. But this is what we have. This is what's reported. It's what was written down on the death certificates, what was written down on the dismissal summaries. I'm sure that with better diagnosis, these numbers would quadruple or more. But what it does tell us that even with this underestimate, that young children have a more severe disease probably than older children, and that adults all the way out to age 80 and probably beyond still can get myocarditis, something that in clinical medicine, most doctors in adult medicine don't think about it. If somebody comes in with a heart attack, quote unquote, at age 60, they assume it's due to blocked arteries. They never think, oh, it could be myocarditis. It's a disease of young people. And that raises the next issue of when do physicians and when do the public in general, people who are not affected by this, think about myocarditis. And it's when there's a sudden death. It's when the athlete dies on the basketball court. It's when, in this case, Rod Stewart's wife went public with her episode of myocarditis, which was in New York, treated at Cornell. And she survived, and she shared her story. And we hope that she will share her story more with us on the website, and we'll be able to incorporate her as more of a spokesperson in the future. But the trouble is that after this happens, there's about a month of interest, and then it fades from memory until the next Randy Travis or famous person. And that's human nature. But our job in this organization is to keep people aware and keep them remembering, even between these episodes. This is a busy slide, but it was published 1 week ago. And I wanted to show you, this is data from the NCAA athletes' sudden death registry. It's a detailed registry. It's well funded. These people have staff. They're able to follow up cases of NCAA college athletes who died. And it's tragic, but it's true that the number one confirmed cause of death is suicide in young athletes, followed by drug use, trauma. Cardiovascular confirmed altogether was very common. And in almost all series, hypertrophic cardiomyopathy, which is a gross description of heart pathology, you can make it without a microscope, is always number one. Anomalous coronary arteries, which are inherited congenital abnormalities, is usually number two. Again, a diagnosis you do not need a microscope for. And then there's a host of things. Myocarditis came way down here. Usually myocarditis is number 3. But the biggest group, no autopsy, but presumed cardiovascular. And 16 not included because they didn't have an autopsy or other data. 35 of these athletes died with no diagnosis. And that's our mission. I'm convinced that some of those 35 young people who died suddenly of myocarditis. And you will never know that if you didn't do a proper autopsy. That's just to illustrate what our mission is here. To give a little bit of historical perspective about myocarditis, the disease was first diagnosed by a pathologist in 1837, Somberheim in Berlin. And it's interesting that today, Berlin is one of the places where they still study myocarditis, perhaps more than anywhere else in the world. Relevant to this group, giant cell myocarditis was described by a different pathologist, that's him, Dr. Saltikow, when he was in Basel, Switzerland in 1905. And he described this is a slide, a microscope slide, and that is a giant cell right there with all the nuclei. And this is a lot of inflammation, these little small blue dots. And they And he's the fellow who described it first in a single case report of a young man who had an abscess and developed fulminant heart failure after the abscess and died of giant cell myocarditis. Perhaps it was some trigger from that acute inflammation. But it's really not giant cell, which is autoimmune, but viral myocarditis that kills most people. And probably the most common virus in the world is influenza in people. And the first case of any viral myocarditis was in 1919 when they described the 1918 pandemic. Famous pandemic killed tens of millions of people worldwide, mostly from pneumonia. But they described myocarditis as a result of influenza in those cases. And then throughout the 40s and 50s it continued. I won't tell any more about the viral story, except to say that in North America, it's still the most commonly identified cause, and it is important. The challenge is unfortunately that there are just so many different viruses that can do it, and the vast majority of people with these viruses are not sick. But when there is a bad influenza going through, in studies from Japan and elsewhere, about 1 in 10 people with bad influenza going to the hospital will have a rise in cardiac troponin, a blood test that illustrates damage to heart muscle cells. Or if you do electrocardiograms, they'll have abnormalities that suggest cardiac injury. So it's pretty common in certain bad strains of influenza. But of course, we don't do heart biopsies on everybody. Heart biopsies carry with them a 1 in 1,000 chance of death and a 1 in 250 chance of perforating the heart. And so if you did thousands of heart biopsies, you'd cause more harm than you would do good. You have to biopsy selectively. And so one of the ways we decide who has myocarditis is not with a biopsy, but with an MRI. And this is the kind of finding, this white line here, this is the heart, the left ventricle, cut in it as a circle. So you open it up and it just looks like a circle of muscle. And black is normal, blood is gray, and that white line is myocarditis. It looks different than a heart attack, which is on the inside of the circle. This is on the outside of the circle. And we know from Lisa's work and others, the reason is that this is a mouse heart, that these blue dots here, right there, that's inflammation, and it's myocarditis, and it's very focal. It's on the outside of the heart where the pericardium is, that's the outer lining of the heart. It's not on the inside or the middle, although it can be. And this slide teaches us something. It teaches us that if you were to do a heart biopsy in most people with myocarditis, you would miss the myocarditis because it's very focal. It could be only 5 or 10% of the heart, but that's all you need to die. All you need to die is a small area, and I'll show you why, because of how the inflammation disrupts electrical conductivity. When I reviewed the world's literature on sudden death in myocarditis, 40% of the people who died who had autopsies and confirmed myocarditis had globally normal hearts. If you'd done an echocardiogram the hour before they died, it probably would have looked quite normal, which is a very sobering fact that just echo-based imaging is not enough to make this diagnosis. We need, as a group, to discover new blood tests, new imaging tools that are more sensitive and specific that can be used in certain populations, perhaps young children with flu-like illnesses, that will allow us to make the probable diagnosis and impact their care. So again, I showed this just to show you that it's focal, it can be often epicardial. If you did a heart biopsy, it would be right here on the inside of the heart, and you would look under the microscope and it would look normal. And you'd be wrong. You'd think it was a normal heart, but it wouldn't be. So I only have 2 other slides because the topic of this talk was supposed to be what is myocarditis. And the message I'm trying to give is that the pathologists say it looks like blue dots under the microscope, and that's true, but it doesn't help you clinically very often because the microscope's often wrong because of sampling error. And if you're an imager, it could be a white spot on an MRI, but again, you have to have access to an MRI to do that test. And our current imaging tools are relatively insensitive. 1 out of 5 people who actually has myocarditis will have a negative MRI because the spatial resolution is is not that great for this disease. But the last two slides talk about the future. One of the people we funded to do research in San Diego, from the Myocarditis Foundation, looked at Coxsackie virus. That's a virus that's very well known to cause inflammation in the heart. And it's named after a town on the Hudson River Valley of Coxsackie, New York, where the virus was first isolated. And the fellow who we funded studied this molecule. It's called dystrophin. It's very important in the structure of heart muscle cells. Heart muscle cells are very contract. They move, the whole cells move in and out like this. And that's how the heart beats. And they move because a couple of actin and myosin move back and forth across each other up here. But that whole complex is connected to the outer part of the cell by this molecule called dystrophin. And he worked on the mechanisms by which Coxsackie virus cleaves dystrophin. And so, independent of any immune response, this virus can cause a cardiomyopathy and arrhythmias by disrupting that molecule. And I show that not to teach molecular biology, but to say that we are working today on the mechanisms of this disease because it's possible, by looking at cleavage fragments of this molecule, that you can diagnose with a simple blood test Coxsackie viral myocarditis without the need to do a heart biopsy. That would be one of the implications of the research that we funded. And to finish, the second to last slide is on arrhythmias. Because many in the room have experienced the loss of someone from sudden death, I wanted to talk for just a moment about what we've been doing to figure out the cause of the arrhythmias. This was in a patient. These are two patients actually with giant cell myocarditis. As you may know, giant cell myocarditis has a very high rate of ventricular tachycardia, and we don't know why. We know there's a lot of inflammation, but we don't know why that kind of inflammation leads to cardiac arrhythmias. And if we knew, we could go back to patients who have viral myocarditis and say maybe it's the same mechanism that makes the people who died of sudden death different than the people who had the same inflammation that didn't die. And so what we looked at in giant cell patients were these molecules called plakoglobin, desmoplakin, plakoglobin. The names don't matter, but in the normal patients, they live happily in the boundary between two muscle cells. They're called desmosomal proteins. And in the giant cell patients, and in other patients who had a disease called cardiac sarcoidosis and a disease called arrhythmogenic right ventricular dysplasia, which both have a lot of arrhythmias, they were very much disrupted. And we figured out that disruption, you see there are nice white lines here, and there are no white lines here, they're disrupted. And we figured out that change was related to local cytokines, IL-17, interleukin-17, and similar cytokines, which led to, in cell cultures, that disruption. And if you were to block those cytokines, you could potentially block the arrhythmogenic pathway. And that would be relevant, because those are drugs that are currently in phase 3 clinical trials that are human-compatible drugs. So potentially, we would have an intervention that would directly impact your risk of arrhythmias based upon the mechanism of cell disruption that we learned from those 1 in a million people who have giant cell. So Candace's experience, which involved a lot of arrhythmias, and others' very, very rare experience with these 1% of myocarditis is giant cell, but that we're learning from that group things that are relevant to everybody else with more common forms. And the last thing, this is on the 10th floor of the building I work in, the Ganda building. His name's Ed. He's a nice guy. They're nurses. And I show this because this is a cell separator and this is a plasmapheresis machine. And we're working on new therapies. It's just an illustrative slide. It happens to be a slide of immunoabsorption, which we did in a couple of clinical trials. But the point is that we're trying actively through these translational studies with Lisa Fairweather, Madeline Cunningham, and others in our work, and the work we fund in 15 labs so far, outside of the direct foundation of investigators, is to learn how ultimately to prevent and treat disease. That's always, when we review grants as a board, after the scientific review, when we're thinking about funding grants, it's always what is likely going to lead to advance treat disease. That's always, when we review grants as a board, after the scientific review, when we're thinking about funding grants, it's always what is likely going to lead to advances in clinical care in the 5 to 10 year time frame. We're not trying to fund fundamental science. We want to fund things that will make a difference in lives relatively soon. And so we work on this kind of therapy which removes antibodies which can cause heart damage. We work on these things called monoclonal antibodies which can be used to target specific pathways that are relevant to arrhythmias or giant cell myocarditis or chronic heart failure. And that's really about what I had to say. 20 minutes. Mostly I'm here to answer your questions. Thanks. Various groups, people are more sophisticated, they read more, there's a higher baseline level of awareness, at least from my perspective, and that would be validated by our website, which is now getting 500,000 hits per month. So we're getting a lot. The growth has been very steady worldwide, and it's not just in the U.S., it's worldwide. It's Lindsay would know the current numbers, but that's approximately.
Lindsay, what are the hits on?
I said, were we up at about 500,000 hits a month? I was for the website.
How many countries?
Yeah, so we're.
Do you know how many countries?
What we need, at least 20,000. So.
Yeah. When you look at unique individuals, it's still approaching 9,000 to 10,000 unique individuals each month.
You said 30 to 40 countries.
From 30 to 40 countries. That keeps growing. So I do believe that reflects a greater awareness.
I'm just going to talk to y'all because I figured trying to explain all these science slides to be a little complicated. So first, I know Leslie wanted me to touch on what we do specifically in our lab and we've kind of been finding, but I wanted to touch on some of the big things that have been happening that have kind of been foundation-wide. And so one thing with what's we're really starting to work on now is we're going to be updating the researchers side of the website. So our website kind of has multiple parts. We've got a great section that Lindsay does an amazing job on that is for all of y'all. But we also want, as we said, we have this mentoring program with the researchers. We also want to provide them an outlet to go onto a forum if they have questions or if they need information or if they need help in order to have this collaboration to help each other and advance the field further. And so that's something that we're really going to be working on this summer, getting that all set up and ready for, we're having a researchers meeting at the Heart Failure Society of America meeting in September in Vegas. And so I'm not sure exactly how many researchers we have thus far, but we've got a number of researchers that are going to be coming, and we're going to have a mini meeting on the side from them going to the heart failure meeting where they'll be able to talk. And we are doing this, it's called a lightning round, where they give, or a 3-minute thesis is the other term for it, where they give one slide and 3 minutes to talk about their research. And we wanted to do this so they all got a chance to see what everyone's doing, to say, well, hey, you were working on this. I think that may help me in what I'm doing. And so we wanted to give everyone an opportunity to kind of present where their research is right now, what their next steps are, so we can help each other keep going further. So that'll be happening in September, so that'll be really exciting. We're also going to have Dr. Cooper there, as well as some other physicians, as well as the PhD and the researchers. And as Lindsay said, me and Leslie were in Australia, and we had a family meeting there, which was amazing. So there was 5 or 6 families and friends of survivors as well as victims that came together at that meeting, and it was really great. It was a great program, and we're trying, in the process of trying to made plans to possibly set up a branch of the Myocarditis Foundation in Australia to allow them to have what we have here, and they'll have family support meetings and things like that. So that'll be a really great next step for the foundation. And but what was really great about this conference is the Myocarditis Foundation co-sponsored a session, or sponsored a session at the conference where some of the top myocarditis physicians presented their research, and there was over 400 people, 400 cardiologists that attended this session that Dr. Cooper moderated. And so he had told me when they first started giving sessions at cardiology conferences, there would be 15 people in the room. And now there's over 400 people coming specifically to this session when they have hundreds of other sessions to choose from. So this is really great for helping build awareness. And I manned the booth when Dr. Cooper was doing his doctor duties. And, we had probably, almost probably 1,000 doctors stop by. I don't know, or it was crazy. At one point, me and Dr. Cooper were at the booth, and we had 7 people actively asking us questions. And about half of the people that we met with, I'd start with, have you heard of myocarditis? And all of them are cardiologists, and about half knew, were very understood and knew the mechanism of myocarditis, and half wanted more information. And they took the pamphlets that we had made up that were for either adult or child cardiologists to help with diagnosis and management of the disease. So it was a great conference. It was very, very productive. So that was Australia. And 2 days later, as she mentioned, I went to Korea with my boss, who is currently in Germany, doing exactly what we did in Korea 2 weeks ago. And we went with Leslie. He luckily invited us. They were having a Mayo Clinic, Samsung Medical Center collaboration, and one of the doctors there is a myocarditis expert in Korea. And he invited us, and we were able to go analyze, I think, about 60 to 70 patient samples that he had there. For a specific marker that we're looking at right now to be a biomarker for heart failure. So it is actually in the line. It is actually not just something that, this means they have myocarditis. It actually is part of the disease process, and it's called soluble ST2. And we found here in the United States, the type of myocarditis we have, it's more prevalent in men. It's higher in men, which with men going to heart failure more often is very indicative of the disease process. But there is a specific type of myocarditis that is seen here in the U.S., but is not as prevalent, that is very prevalent in Korea, called fulminant myocarditis, that is normally very, with my basic clinical understanding, is very severe and always goes to heart failure. And we found that the ST2 levels in these fulminant patients was 100 times higher than the non-fulminant, where patients normally recover. And so that was very exciting for a biomarker, and we found that it's actually higher in females. So this could be very interesting for predicting what patients could go on to heart failure. If you look at this level, you diagnose myocarditis, and if we can look at this level of this biomarker in the blood, it's just a blood biomarker, we could predict who's going to have worse disease, who could go into heart failure, who could pass away. So that was really exciting, and we are now developing an animal model to model that and figure out the mechanism of this specific type of myocarditis in animals, as well as developing an animal model for giant cell myocarditis, because right now there is not a good animal model. And Delisa believes we may have a mechanism to induce that. So that'll be very great for figuring out the pathogenesis of the disease and eventually the cure. The last main big point foundation-wise is we are currently enrolling and recruiting for the biobank that the foundation has wrote a grant for, has received the grant, or maybe, but we're doing it, So currently in the U.S., the only site that is recruiting is at Mayo Clinic, but we have plans to extend that to other sites throughout the U.S. So a patient that ends up being diagnosed with myocarditis, perimyocarditis, or postpartum cardiomyopathy at Mayo Clinic will be enrolled into the study, and will have blood drawn, and that blood will be shipped overnight to my lab and Delisa's lab her lab, and Baltimore at Johns Hopkins, and we will isolate the immune cells and the sera from these blood samples, and then we bank the biological samples till we have enough samples here that we're able to actually analyze and figure out mechanisms. And this is the kind of thing that they had in Korea, and that's why we went there, because they had 60 samples that they had already collected that we could go and do the analysis at one time. So that's our goal, having that here. And Delise is currently in Germany. Who knows where we are these days. She's currently in Germany analyzing, I think, over 100 patient samples there for this same marker, so that we have enough patients to be able to publish a paper to then really get this out there as a potential marker that can be looked at in the future. So there's the site in Czech Republic and Germany that have already started recruiting, and we're almost ready to start at the South Africa site. And we are in the process of setting up a site in Italy for perimyocarditis. And there are future sites in Germany and the US that we are in the process of working on. So this will be really great for finding out when we see something as an important mechanism in the mouse, we can look at it in these patients and make sure what we're doing in the mice translates to patients. Because it doesn't matter what you can find a whole mechanism in a mouse, but if it doesn't mean anything for treating and understanding diagnosis in patients, if there's no point. So that's what's really exciting going on with the foundation. And I was didn't want to really talk about our specific research as much, but I'll just mention, because Leslie was talking about how we do really study sex differences and why men have more severe disease, why they progress. And one of the just very basic things that we've just finished working on, and some of it is published, is looking at how testosterone increases myocarditis and estrogen decreases myocarditis. So what we do is a very basic understanding is we have mice and we remove the gonads, or the testes, from male mice, which essentially removes all of their testosterone. And when you do that, a normal male mouse with myocarditis has about 80% inflammation. So he showed you that slide about the blue dots. You measure how much blue dots there is over how much whole heart there is. So that's about 80%. So 80% of the hearts in these mice is full of inflammation in these male mice. When you remove the gonads, you remove the testosterone, the disease goes down to 20%. When we then have a pellet full of testosterone powder that we implant in the back of the neck of these mice, and when we do that, then it replenishes the testosterone level in these mice, and then the inflammation goes back up to 80%. So this is suggesting that testosterone is driving the inflammation in these mice. So this could be a direct mechanism for why men have more severe disease. And in females, that has been published by Michael Coronado, Delise's previous PhD student. When I came to the lab, one of the first things I did, which is really hard, is ovarectomies, where you take the ovaries out of a mouse to remove its estrogen. So a normal female mouse has about 20% inflammation. So it has just a normal inflammation is a lot lower than a male mouse. If you remove the ovaries, remove the estrogen, the disease increases to 60 to 80%. You give an estrogen pellet to replace the estrogen in these female mice, it goes back down to 20%. So this suggests that estrogen is playing an active role in keeping the inflammation lower in the females. So these are things that we're now looking at, the little mechanisms that we know are very important in myocarditis, and seeing if estrogen is specifically driving those things, and could be why males have higher disease and progress to dilated cardiomyopathy. So there's lots of things that we're working on, and then my specific projects are looking at vitamin D, which most people are unaware is actually a hormone, and looking at endocrine disruptors. So I'm sure many of you have heard of BPA. In plastic, they say, oh, all your water bottles are BPA free now. So that's my project, one of my main projects I'm working on right now. But then people say, well, they're taking it out of water bottles, so why are you studying it? It's not important. They're replacing it with a compound called BPS, or bisphenol S, which is just very slightly different and believed to be the same kind of compound that could cause the same effects. So these are the projects we're kind of working on, as well as another project on ozone and pollution, and how ozone and pollution could increase myocarditis. So those are kind of the main projects, both the foundation and overview of kind of our specific clinical translational studies as well as what we're working on in our lab right now. Okay. Thank you.